can you tell us a little bit more about your particular area of research and what drove you or attracted you to that part of the field?
Sure. So I certainly, when I started my lab, was not intentionally moving into the stem cell and gene therapy field. I'm actually, by pedigree, I studied how cells respond to virus, the cytokines they release, the way they can fight off virus infection. And when I started my own lab, I branched out a little bit and started investigating a field of small RNAs. They're generally called microRNAs, and every cell of the mammalian body, and in fact all eukaryotic species, produce these little RNAs, and they fine-tune regulation, or fine-tune transcription rather. And I started investigating the interplay between the cell response to virus infection and these small RNAs, and it opened up this world that was very unpredictable, but it led to two discoveries that I think bring me here today. The first is that you can exploit the expression of those small RNAs to control where a virus or a vector can and cannot go, which has a lot of value in the gene therapy and virus oncolytic field in general. And we made a serendipitous discovery that you can engineer different RNA viruses or virus-like particles to produce these small RNAs, which provides tremendous potential to the field of RNAi therapeutics. So RNAi therapeutics are these two duplex RNAs that were discovered 10, 15 years ago by Tom Tuschl and many others. It's a Nobel Prize-winning discovery, but it had billions of dollars invested into it. The industry boomed, and the thought was you could cure anything if you just knew what gene to target. And while the potential of siRNAs are still tremendous, what seems to be a barrier that can't be overcome is the delivery. And so what my work touches on is a novel approach towards delivery of those siRNAs.
Has there been a particular mentor or mentors along the pathway that's kind of helped you?
Sure. No, it's a great question actually, and I think it's kind of nice to reminisce and look back on how all of my mentors or colleagues have influenced my lab, how I run my lab, and the way it's set up today. So in my PhD, my mentor should get credit for introducing me to the field of oncolytics. The use of using viruses to kill cancer is an amazing approach, and not one that I think, at least at the time, was widely known. So he certainly kind of put viruses as therapeutics on my radar. My postdoctoral mentor, his name is Tom Iniadis. He was at Harvard at the time, now at Columbia. He, I just think, demonstrated to me that good science begets more good science, and you just follow the results. And he was just such a great leader and role model that I probably give him the most credit for who I am today and for the success of my lab. But I also right now have a great chair. He's a member of the National Academy of Science. His name is Peter Palese. He also has fantastic attributes that really demonstrate, again, that good science begets better and more good science, as well as the connection, I think, between transforming results in the lab to therapeutics that can be translated into everyday medicine. The cliche from bench to bedside is actually, in fact, possible, especially in the world of vector design and vector engineering and vaccine development, which is kind of all encompassed in the work that I do.
What does winning this award or being recognized for this award mean to you?
It means a lot to me. An award like this, especially coming from a society that I wasn't directly involved in per se, was especially meaningful because it, one, tells me that my work is transformative and I'm crossing fields. But for someone like me, a young investigator, I think it's critical to provide, a little bit of reassuring that the research that I do is multidisciplinary, that I'm on the right track, that the research group I have is strong, and it encourages me to keep on developing, following the data that I have, and developing the program that I have. And I think especially as a young investigator, that kind of confidence is really gained from awards like this.
You mentioned being recognized that your research is being transformative. Is there, looking ahead, are there other contributions you hope to make aside from that, or within the society as a whole?
Oh, I certainly hope so. So, to be honest with you, 5 years ago, I would never have predicted in a million years that I would be doing the research that I'm doing and sitting here having won an award from American Society for Gene and Cell Therapy. Like that was difficult to predict, and I certainly hope that 5 years from now, my work has again, continued to expand and followed new applications. I do have a great interest in developing what we presently are building on. The platform itself is incredibly powerful. We have the ability to now make a vector that is designed to go to a very, very specific tissue and deliver a very, very specific payload of whatever you want to put into that vector. And that is unheard of potential in the field right now. And so I hope to develop it further and prove that it actually has the promise that, or can deliver on the promise that it actually has right now.
How has ASGCT contributed to your research as a whole, from the society perspective?
So I think the best way to answer that question is really just, it's meetings like this that really do that. So this is my 3rd meeting now, and it's meetings like this that really demonstrate the breadth of the field, where people are going in the field. It's a great place to initiate collaborations, and it's actually a really collaborative group coming from the innate immunity camp of scientists, which gets very aggressive and competitive. I really like this community as one that really does seem to be doing research for the greater good and hopes to generate improved therapeutics. And certainly I'm a huge fan of not just gene therapy, but virus oncolytics. And I certainly hope that in my lifetime I see these things push past phase 3 clinical trials and start being used in everyday clinics. So I think the society provides a great service to the community.